Good afternoon. My name is Ali Rezania. I'm a CSO of Satellite. Delighted to be here to share with you the pioneering work we're doing at Satellite regarding next-gen tissue therapeutics. So Satellite is an emerging preclinical stage company. We were founded just a couple of years ago. We came out of stealth mode last year. Let's see if I can figure this one out. Okay. So our mission is to restore hope to patients and their families suffering from severe, life-threatening conditions. And our vision is to lead the next-gen region med by establishing tissue therapeutics as a new pillar of medicine with a laser focus on liver disease. So we've assembled a very talented and experienced team at Satellite. This is based on foundational work from our founders lab, Sanjita Bhatia from MIT and Christian from BU. We've been very lucky to be backed by a premier investor syndicate, leading to a healthy Series A raise last year. And we've also assembled a very experienced, knowledgeable advisory team that we tap into across many functions, whether it's science, BD, finance, and operations. So the foundational work has been laid by our founders over a 20-year span. So this is not a result of one or two papers. It's a result of many seminal papers that are highlighted here, reflecting the complexity of making tissue therapeutics come to reality. So the work encompasses addressing areas such as bioengineering, deep knowledge of just liver biology, as well as tissue therapeutics itself. So what is the basic technology? So we refer to technology as satellite adapted tissue, SAT for short. So this cartoon is supposed to highlight, it's a simple cartoon trying to highlight what actually the technology is about, but actually executing it is very difficult. So we work on what we call highly functional expanded solid organ-derived cells. So these are not iPS-derived cells. And they're combined with stromal cells or nursing cells that gives rise to what we call seeds or aggregates. The stromal cells are essential for the maintenance of the functionality and durability of the effector cells post-transplant. So if we just transplant the expanded derived cells, they don't function long-term. And this is a really important element, which is what has stymied the field for a long time. And as well, we've also found empirically that modularity in the size and distribution of these cellular components has a significant impact on the in vivo outcomes. So the takeaway is micro architecture matters and enables the delivery of a tissue therapeutics that would have been proven otherwise to be very difficult to do just with one cell type. So our focus is liver disease. So invariably, severe liver disease leads to the demise of hepatocytes, the main parenchymal cells in the liver. This insult can take the form of, for example, drug-mediated impact, infections, hereditary genetic disorders, or alcohol or diet-induced liver disease. Regardless, the current interventions do little to stop the progression of severe liver disease, leading to high mortality. And this is an area that we are focused on. So treatment of severe liver disease, I'm highlighting two of the options that has been around for 30, 50 years. One is orthotopic liver transplant. It does work. However, it suffers from some obvious drawbacks. First, there are not that many organs to go around. So there's a long waiting list, unfortunately, for a lot of the patients. Also carries significant surgical risk. There's a lot of infections as well as bleeding. So operationally, you can easily envision that taking the parenchymal cells out of the liver and transplanting those, it becomes much easier and safer, less invasive, and less costly. And indeed, that's the case for hepatocyte transplantation. However, this treatment has not gained wide clinical use. It's only been 100+ cases worldwide. And the main reasons are cited there. And again, they're obvious. There's these cells, you need a large mass of cells. A typical adult human liver has over 200 billion hepatocytes. Even if you need a fraction of them, that translates to a large cell mass. The cells don't survive well post-isolation, and also they just don't engraft well post-engraftment, regardless of what ectopic site you put these in. And there's a checkered history of this if you read the literature regarding hepatocyte transplantation. So our aim is using our technology to address these gaps. So at Satellite, we're developing 4 different technologies to address these gaps. One is expansion of hepatocytes. So we figured out how to expand adult hepatocytes. We can readily now expand them over 1,000-fold. Seed formation is the other element of the technology, where you have to combine these expanded cells and matured cell with a nursing cell to enable in vivo engraftment as well as durability. And as well as combine the whole thing to doing in a scalable fashion, so industrializing the whole process of expansion and seed formation. And lastly, recently we also embarked on engineering of these expanded cells. To our surprise, these cells are actually fairly easy to engineer. I come from a genetic engineering background, and I was skeptical when I joined Satellite that you could engineer these primary hepatocytes, and indeed that was the case, but the expanded cells is a different story. Hepatocytes are well known. They're amazing biofactory of secreted protein. Average human hepatocyte secretes over 10 million molecules of albumin per minute. No cell type in your body comes close to that level of secretory capacity. So we're trying to leverage that to areas outside liver disease as well. Okay, so what does manufacturing look like? So we spent a lot of time on this whole process. So it starts with isolation from the donor liver. This was not an easy thing to do, so identifying the CQAs and CPPs that enable robust hepatocyte isolation was not an easy thing to establish, but now we have it in hand. We've done this over a dozen times now, where we establish a bank that's thoroughly characterized, and then that bank is further expanded. So as I said, we can easily expand these 1,000+ fold. And then the cells are further matured and then combined with the GMP stromal cells. Now we have access to multiple GMP banks that enable this combination to make the seeds or aggregates. The aggregates and seeds are also, we can freeze them and then thaw them post-thaw with a high viability, which I will show you in a second. So what does expansion and maturation look like? So we start with very low density cultures shown on the left-hand panel. These are almost clonal level. They expand well over 1,000-fold. This expansion process requires the EMT, so that cells go through an epithelial mesenchymal transition. So they lose a lot of their properties of hepatocytes post-expansion. And you can see that in the middle panel, we do a thorough characterization of these cells, both by genetic markers as well as by protein. So in the middle panel, we do what we call the gene scorecard, looking at transcription factors that are hallmarks of hepatocytes. And post-expansion in yellow, there's a significant precipitous drop post-expansion. However, we put these cells post-expansion in a maturation media, about 4 or 5 days. They regain back all of those transcription factors. So transcription factors by itself is not enough. Obviously, hepatocytes perform a lot of different functions, and one of them is urea cycle enzymes. And we look at those and highlight it here. Post-expansion and maturation, we regain a lot of those markers back at the transcript level. So about protein, I'm highlighting here 2 of the most secreted protein by hepatocytes, albumin and A1AT, and we regain a significant amount of those secretory capacity post-maturation as well. Another important element of hepatocytes is their drug metabolism. So we look at CYP activity. Post-expansion, that's CYP activity when the cells are exposed to a drug like rifampicin, for example. You can see baseline to induced, there's not much difference. However, when we mature these cells, comparing post-maturation at the baseline versus induced, there's a significant uptake in CYP enzyme activity, very much mirroring their sourced starting primary human hepatocytes. As I said in the previous slides, important element of product development, especially for cell therapy, is the ability to freeze and thaw the cells. So we spent almost a year on this trying to figure out, because here we have 2 different cell types, it's not only one cell type, how we can have really good viability post-thaw. And now we're getting very similar to what we see as compared to fresh cultures. Just having viability by itself is not enough. You also need to look at function. So one of the elements we look at is, for example, ammonia clearance, a reflection of the urea cycle activity, comparing fresh seeds compared to frozen. And now we have this very similar activity. And this serves as a very nice potency assay as a measure of how well functioning your cells are post-thaw. So that was in vitro. What about in vivo? In vivo, we use a plethora of different preclinical models. On the left, you're showing preclinical model we use for looking at engraftment. So these are immune-compromised animals. We transplant these cells, these seeds in these animals and track them as a function of time and just measure secretory proteins. So for example, here I'm showing albumin. We see amazing levels of albumin being secreted by these cells. And the takeaway here is that it is durable, right? So a lot of other teams have shown this, that you can do this, but the durability has been subpar. So that's in a small animal, a 25-gram mice. When we go to a 10-kilogram mini pig, we also were delighted to see that, again, this is in a xeno model, so the animals receive immune suppression. So we can look at prolonged survival because the cells eventually will get rejected. However, we were delighted to see at the very early time point, 3 days to 10 days, we see significant levels of albumin transferrin A1AT secreted by these cells using a fairly low dose of cells. Again, this is enabled by the seed approach. If you had done this just with the cells, hepatocyte itself, we don't get this durability. So this is what the graphs look when we take them out. This is only 2 weeks post-transplant in the mouse model. These are confocal images. The green marks the hepatocytes, and the host blood vasculature is marked with red color. And we were very excited to see that the fenestration of blood vessels invading the hepatocytes, very much mirroring what you see in the natural liver architecture. Again, that juxtaposition and close proximity of blood vessels to the hepatic structures is key for the durability of function. So this we see both in mouse as well as in pigs. So in an acute liver failure model, this is one of the animal models that have been around for a while. It's referred to as TK-NOX, it's an immune-compromised mouse. The hepatocytes have a transgene in them under albumin promoter that they express thymidine kinase. You give the animals toxin. In this case, we give 3 rounds of toxin. The cells that have the transgene essentially phosphorylate the drug, and then they apoptose. So the host hepatocytes essentially fade away, they go away, and then as a result, the animals essentially die out. And we see that in that TK-NOG that have received the toxin 3 rounds, the animals don't survive past 50 days. However, in the same animals where we give these seeds and transplant these seeds in, we see a significant number of animals surviving. When we take the grafts out and we stain them for markers unique to adult hepatocytes, for example, here we staining them with CPS1, carbamylophosphatase synthase, which is the first major mitochondrial ureal cycle enzymes that's only expressed highly in mature hepatocytes. I mean, we're very excited to see that indeed all the cells have this marker expressed. So what we're showing with this model is that we can demonstrate metabolic offloading to promote native organ repair as well. So what's next for us? So Satellite is positioned to build out a robust clinical pipeline in advanced liver disease over the next 3 to 5 years. We are aiming to declare our first product candidate next year. And as I alluded to earlier, we expanded our efforts in cell engineering of expanded hepatocytes. We think this is an untapped territory. And then also we're exploring value-added partnership outside of liver disease. So with that, thank you, and I'll be happy to take questions after the meeting.